Hello, and welcome to the third talk in the Mathematical and Computational Ophthalmology seminar series. My name's Paul Roberts, and I'm a Centre Fellow in the Centre for Systems Modelling and Quantitative Biomedicine at the University of Birmingham, where I work in the mathematical modelling of the eye. I'm the creator and host of this seminar series, together with David Crabb, who heads the Crabb Lab at City University of London, and Phil Luthert, a Professor of Pathology at the UCL Institute of Ophthalmology. This seminar series aims to showcase research in mathematical and computational ophthalmology to the broader ophthalmological, vision science, medical, mathematical and computational communities, raising awareness of the exciting research being conducted in the field and encouraging future work in this area. Do subscribe to this YouTube channel if you'd like to keep up to date on past and future talks. These are also listed on our worldwide page, the link to which can be found below in the About section of this YouTube channel. And our thanks go to Worldwide for instigating this platform for sharing scientific research. Before introducing our speaker, Anna Pandolfi, I would like to remind our listeners that David Crabb, Phil Luthert and I are guest editing a special issue on mathematical and computational ophthalmology in the journal Investigative Ophthalmology and Visual Sciences, or IOVS. It's our hope that this issue will serve to showcase research in mathematical and computational ophthalmology to the broader medical, experimental and quantitative communities, raising the profile of the field and promoting the research of those working within it. We welcome submissions of original research applying quantitative methods to examine any aspect of visual system structure or function. The deadline for submission is the 1st of December 2023. All submissions will be peer reviewed and publication fees will apply to papers that are accepted. A link to the IOVS special issue advert can be found below in the About section of this YouTube channel. So I coined the term mathematical ophthalmology to denote the field which uses mathematical tools to preserve vision by elucidating the mechanisms underpinning ophthalmic health and disease, while computational ophthalmology is an existing term used to denote the field which uses computational algorithms, in particular AI, to preserve vision by identifying and predicting ocular features and risks. Thus, the focus of mathematical ophthalmology is upon elucidating mechanisms, while that of computational ophthalmology is upon prediction and identification. In our last seminar, focusing on computational ophthalmology, Aaron Lee gave a fantastic talk on deep learning applications in ophthalmology, the recording for which can be found below on our YouTube playlist, together with recordings of all previous talks. This week, we're switching back to mathematical ophthalmology with our speaker, Anna Pandolfi. If you'd like to ask Anna a question, then please do send these in via the YouTube chat, both during and after the talk. The talk will last for about 45 minutes, followed by 15 minutes for Q&A, in which I will relay your questions to Anna. So it gives me great pleasure to introduce Anna Pandolfi. Anna is Professor of Structural Mechanics and Solid Mechanics at the Politecnico di Milano, where she first started in 1995 as Assistant Professor. Since 1996, Anna has also held an intermittent visiting associate position at Caltech in the USA. She is the editor-in-chief of the Springer journal Mechanica and serves on the editorial boards of 6 international journals. Anna was an elected member of the Euromet Council from 2016 to 2021 and served as the coordinator of the Italian group of computational mechanics from 2015 to 2019. She is the author or co-author of more than 90 publications in international peer-reviewed journals and of a further 50 scientific works. Her major contributions to scientific research are in the field of computational mechanics, with the development of advanced fracture tracking techniques such as cohesive elements and eigen erosion, particle methods for the discretization of solids and fluids, a new concept concrete with attenuation properties known as metaconcrete, and multi-scale material models for porous brittle materials, known as brittle damage, used to simulate fracking. Anna's second field of intensive research, of particular relevance to today's talk, is biomechanics of soft tissues, applied in particular to the behaviour of active tissues, muscles, intestines, and eyes. So I'll hand over now to Anna to present her talk entitled Computational Models and Experimental Methods for the Human Cornea. Anna.
Can you hear me? Okay.
Yes, it's all good.
Thank you, Paul. First of all, I would like to thank the organizer for inviting me to give this presentation. And I think this is a very great opportunity to share advanced work on computational and mathematical ophthalmology. So my talk is concerning computational models and experimental methods for the human cornea. Of course, speaking at this audience, I am going to tell things that are pretty obvious about the eye. The eye is a refractive system that is able to deviate the light rays onto the retina, and there is a sequence of lenses. The lens that I am more interested in is the cornea, the anterior lens that is a concave convex shell that is able to provide almost 2/3 of the total refractive power of the eye, about 43 diopters. The cornea has this particular shape because it has to withstand the intraocular pressure. In average, the intraocular pressure exerted by the gels that are filling the eye is between 15 and 18 millimeters of mercury. The particular shape of the cornea provides the refractive power that can be computed in an approximate way through the thin lens equation. And this equation that probably every one of you have been seen before contains the geometry of the cornea, in particular the radius of the anterior surface and the radius of the posterior surface. So if you wanted to modify somehow the refractive power of the eye system, you can think to modify the refractive power of the cornea because it's the most accessible part of the eye. So it's, you can think to implement a modification of the total refractive power by changing the geometry of the anterior radius of the cornea. This can be done in several ways. With a reversible system by introducing, for example, small devices in the stroma, in the different layers of the cornea, or it can be done through irreversible procedures performed with a laser. In particular, these procedures have become very popular nowadays. And among them, I would like to mention the three most diffused. The idea is to remove a portion of the corneal tissue with the shape of a small lens by using the laser, by using laser ablation, or by using a cut or creating a cut within the stroma and removing a small lens inside the stroma itself. So I would like to remember the procedure that is called transepithelial or photorefractive keratectomy that applies the laser directly on the anterior surface of the cornea and reprofiles the cornea in order to get the refractive power that one wants to have, or the laser in situ keratomylesis, LASIK, that refers to a procedure where before doing the ablation, the reprofiling of the material, you make with a keratome or with the laser a flap. You create a flap that is moved back and then creates a surface where the laser can act and can, of course, ablate the tissue. Then the flap is put back in the original position. Of course, this procedure requires the cutting of a part of the tissue that forms the cornea. The small lenticle extraction, called briefly SMILE, is a procedure where you cut the small lens using the laser at the interior of the stroma. And then by creating a small lateral cut, you remove with tweezers the small lens. And you obtain something which is very similar to the results of the LASIK, with the difference that instead of having a very long cut that involves almost 3/4 of the circle of the flap, in the SMILE, you have a smaller cut, which is not larger than 1/4 of the total, I would say, circumferential cut that you have instead in the LASIK. There are many advantages by using these procedures. First of all, we have a very advanced totally automatized laser machines that are able to provide a very high precision. So you get a perfect reprofiling in the case the cornea is healthy. But you have also some drawbacks. For example, the surgery is irreversible, so you cannot go back to the previous situation. And especially in the case of a LASIK, you may observe a long-term instability, the so-called ectasia. And furthermore, you cannot use these technologies with unhealthy tissues, like the ones that are affected by the pathology known as keratoconus. So just briefly, because I think everyone knows how is the structure of the cornea. The cornea is not an homogeneous tissue, but is a layered tissue made by different layers with different characteristics. In particular, from the anterior to the posterior side, we identify a cellular layer called the epithelium, which is quite thick, about 50 micron thick. Then we have the Bowman membrane, which is much smaller, and the wide stroma. The stroma is really the main layer of the cornea. Then you have a basal membrane called the Schmidt, and then we have the monolayer endothelium in the posterior side of the cornea. Since from the mechanical point of view, the most thick layer, the stroma, acquires the structural functions, we have been studying for many years the characteristics, the features of the stroma. The stroma is made by a composite material where you have a matrix consisting of elastin and proteoglycans, where you have sheets of collagen fibrils. These sheets are called lamellae, and the lamellae derive from an organization of very highly hierarchical organization of the tropocollagen molecule that form microfibrils. In the picture here, you can see a schematic that shows how the micro molecules of the tropocollagen are connected one with the other in order to form microcables called fibrils. In the second picture, you see the fibrils of a mice eye that are all parallel one with respect to the other. They run in a parallel way, and this is necessary in order to guarantee the passing of the light through the cornea. Otherwise, the cornea would not be transparent. Finally, the third level of the organization is all these little fibrils get together in order to form the sheets called lamellae. And lamellae run in all the direction of the cornea and are interlaced one with the other, as you can appreciate from this picture. The fact is that this interlacing is very evident in the anterior surface of the cornea for about 1/3 of the total thickness, and becomes less relevant in the posterior side. So in the posterior side, you don't have interlacing, you just have the stack of the lamellae one on top of the other. Another observation that can be done in the structure of the collagen fibrils can be obtained by X-ray imaging that show that 40% of the collagen is arranged according to an orthogonal disposition. So we have fibrils that run from the superior to the inferior side of the cornea, or from the nasal to the temporal side. And at the center, they form this kind of orthogonal arrangement. Moreover, in the limbus, in correspondence of the limbus, you have fibrils that run circumferentially. And of course, you also must have some radial fibrils in order to guarantee the formation of the shell. Otherwise, structurally, it will not work. Now, one of the problems that has been observed in big pathologies like ectasia and keratoconus is related to the fact that this very nice organization, hierarchical organization of the collagen fibrils, is missed. In particular, in the keratoconus, one can observe that the spherical shape of the cornea is lost because it can be observed a bulging that is typical of weakened tissues. So if you look, for example, at this image on the right of the slide, it is shown the boundary between an healthy cornea, the top one, and the keratoconic cornea. This is an image taken post-mortem from an eye that has been treated with epikeratoplasty, so a transplant of cornea. And you can see that the donor cornea is very well organized in terms of lamellae that run one parallel to the other, while the receiver cornea is totally disorganized. So this idea that when the structure of the fibrils is lost, you don't have the possibility anymore to exert the right confinement for the internal fluids of the eye, is the basic idea for the formation of the keratoconus. And then to counter, to attack, let's say, to this idea, has been proposed to use a technology that is called cross-linking, therapeutic cross-linking, where you try to reconstruct the chemical bonds that are able to keep all the fibrils together in order to form the lamellae, and all the lamellae together in order to form this stack of lamellae that are typical of the healthy cornea. So after the presentation of the basic ideas about the eyes and the pathology that we are interested in, now I will show you briefly some of the results that have been obtained in years, and some of them are very recent actually. So first of all, I will illustrate a comprehensive geometric and mechanical model of the human cornea that may be adaptable to animal corneas. Then I will show some simulation of a therapeutic of cross-linking. First of all, I will show the experiment that in our group have been performing on pig corneas. And then I will show how we were able to model the stiffening caused by the cross-linking. Then I will show some models of transepithelial, LASIK, and SBILE refractive surgery. And finally, I will show how we try to model the formation of ectasion and keratoconus in the form of continuum models based on finite elements and in terms of discrete models. And then I will conclude with some work in progress and the acknowledgements of my collaborators. So the modern machines, the diagnostic machines that are used in ophthalmology, they are able to provide the images very detailed of all the structure of the eyes. And in particular, the cornea is the one that has been more studied and is more precise than any other structure. So from the images, you can get the geometry of the anterior surface of the cornea and the posterior surface of the cornea in terms of collection of points. And this is different by patient to patient. So every patient will have a different geometry. So from this collection of points, we are able, through some mathematical code that we developed in years, to reconstruct the patient-specific geometry of the cornea for the particular patient that we are interested in considering. Of course, this is a model, and we have to account for many problems. For example, the boundary conditions. We are just modeling the cornea. We are disregarding the presence of the sclera and all the other tissues that are surrounding the cornea. So we have to provide suitable boundary conditions in order to mimic the presence or the absence of other tissues. Then the intraocular pressure of the patient is unknown. Okay, it's known, but we probably are all sure that the measurements of the IOP is affected by the fact that is an indirect measurement. So you go through a tissue, which is the corneal tissue, in order to get this information. Moreover, the images that we get of the cornea are images of the tissue that is reacting to the intraocular pressure, so it's deformed. And in order to have a good mathematical model of the cornea, you have to recover the unstressed geometry. So all these issues have been afforded in our models. And from the geometrical point of view, we say we are pretty in a good situation. And the other important aspect is the modeling of the material. For the stroma, I'm considering at this moment only the stroma because given its dominant thickness, the other layers can be disregarded in the mechanical consideration of the behavior of the cornea. For the description of the mechanical model, the constitutive model of the cornea, we make a recursion to hyperelasticity, an hyperelastic material model that accounts for the volumetric deformation, for the deformation that are related to the portion of the material which behaves isotropically, and then a contribution that comes from the portion of material that is instead characterized by the fibers. So is really anisotropic. It is the composition of the energy density, the strain energy density in additive form is quite usual in all the biomechanics of the soft tissues. I'm interested in speaking about the contribution that comes for the anisotropy. We started to look up the works done by Spencer, Itkov, and Neff that were considering the anisotropic behavior in terms of invariance by considering tensors that describe the structure. For example, if M is the orientation of the fiber, you can construct the structure tensor by the idea, the M dyad M, and you contract this into the Cauchy-Green tensors, excuse me. And so this is a scalar, and you can try to describe the behavior of the material by introducing this scalar. And the idea to model the distribution of the fibrils in this way was proposed by Holzapfel and Gasser. And we inherited this idea. And with Marcello Basta, in a few years later, we tried to characterize better the distribution of the fibrils in order to account for the fact that you have a variability between individual and variability between the location of the fibrils. So we characterize this material model not only with an average orientation of the fibrils, but also with a variance. And we've been using this variance model of the fibrils in all our applications. So at the end, we come out with a model of the eye that is pretty strong from the geometrical point of view, that accounts for the presence of the fibrils, and these fibrils are characterized by a certain level of stochasticity. We did several simulations of the inflation test, of pressurization test, the air path test, indentation test, by using this model. I'm not speaking about that because I would like to present the most recent results that we obtained with our group. So recent works concern the modeling of the cross-linking. And of course, we wanted to have some data that we were able to deal with a complete information. So we decided to produce an experimental campaign on pig corneas. And let's just recall a little bit what is the cross-linking. The APO for cross-linking protocol has been proposed by Wollensack, Sperl, and Siller in 2003, and it requires the elimination of the epithelium with a scalpel, the inhibition of the stroma with drops of riboflavin A, which is a kind of a vitamin, and then the irradiation of the exposed surface of the cornea to ultraviolet rays. And the protocol, the original protocol proposed by Wollensack and collaborators was lasting for 60 minutes, a very long time. Recently, the protocol has been modified and reduced to 20 minutes. The important thing is to keep the amount of energy that is totally delivered on the surface equal. And then you can increase the power of the radiation in order to get the same amount of energy in a smaller time, in a reduced time. So we followed the accelerated protocol proposed by our colleague Cosimo Mazzotta on the pig's cornea. Very quickly here, I would like to show this image that shows collagen fibrils that are weakened because there is not a sufficient presence of linkers of bonds between the fibrillae. And after the inhibition with riboflavin and the irradiation with ultraviolet rays, you observe the formation of free oxygen radicals that favor the formation of new links, new cross links that keep together the lamellae on the same at different levels, and the fibrils on the same lamella. So with our study, with our experimental and numerical study, we want to see if we are able to predict the increment of the local stiffness if you change, if you modify the duration of the irradiation. Okay, so we have we use the same machine that had the same power, the same irradiation power, and we want to see what is the effect of incrementing the duration of the irradiation in terms of local stiffness increment, and this in terms also of penetration of the reinforcement into the cornea thickness. So we divided our 90 corneas, pig corneas, in 5 groups. Every group had a certain number of corneas and a different irradiation time, 2.5, 5, 10, 15, and 20 minutes. For every group, for every cornea, we took measurements of the geometry. And the measurements were not, of course, done with the advanced machines that are used for humans. They were made on taking pictures, accurate pictures of the cornea, and measuring the radius, the diameter, the thickness, and all this information. And by modifying our code for the generation of the cornea, we were able to reconstruct the geometry of the pig corneas. We didn't do this for the 90 corneas. Of course, we evaluated the average measurements for every group, and we built up 5 different corneas for the models using the average measurements. Okay, so we did our experiments in this way. After removing the epithelium, we put the, I have to go back 1 second, sorry, because this picture was not shown before. Here, we put the pig cornea into a particular experimental rig. The cornea was pressurized from behind, and the pressure was growing from 3 millimeters of mercury to 30 millimeters of mercury, and we measured the change of the shape of the cornea. Okay? So we did that for all the corneas. Here you can see a group of results that have been done for the 2.5 minutes of irradiation. Then we removed the pressure. We did the cross-linking, and we increased again the pressure, bringing back to 30 millimeters of mercury the intraocular pressure of the eye. And here you can see for the group of 2.5 minutes the results with the average and the dispersion. In the picture that is on the right, you see instead the average, only the average curves for the 5 groups. In the pre-cross-linking case and the post-cross-linking case. The curves show a very clear, a very evident increase of the stiffness. But what we also observe is that we have dispersion of the behavior among the 5 groups. So it's not really easy to evaluate the increment of the stiffness for each group because the starting conditions are very different. in order to evaluate in the best way that we could at the time the increment of the stiffness due to cross-linking, we made use of the linearized shell theory in order to estimate the average secant elastic modulus of the corneas. And of course, we take a measurement only for instead of considering the variability that you can have in every point of the cornea. So we get one number. We did the average for all the corneas belonging to the same group, and we found that the average secant modulus for every group. And the white blocks in the picture, they show the average secant modulus for the group using the linearized shell theory. You can see that there is a big dispersion in the values according to the group. So there is not the same stiffness at the beginning. Then we did the same with the treated cornea, and we got the gray bars. So you can see that in all the cases, you have an increase, a very big increase in some cases of the stiffness. And the interesting thing is that the dispersion of the results is contained. So the higher is the duration time, the smaller is the dispersion that you get in the postoperative cases. We tried to do some simulation. I'm speaking about a couple of years ago, some simulation in order to see if it were possible to estimate the increase of the stiffness from the single group results, but we failed. It was not possible in the sense that we were not able to find a unique number, a unique coefficient of amplification of the stiffness that was able to return, starting from the preoperative stiffness, to return the values of the postoperative stiffness. We had to do, let's say, a calibration of the material properties before and after the surgery. That was not really exciting. But recently, with Alessandra Bonfanti, we did something interesting that seems to be instead working. Because we make the assumption that the original stiffness of the cornea is varying linearly. Things that has been observed clinically, so it's not something that is invented. And then we decided that we have to try to guess the way the irradiation through cross-linking affects the stiffness of the cornea. So we made three different assumptions. The first assumption is that the cross-linking increases the stiffness of the cornea linearly. So the maximum increase is at the anterior surface, and there is a null increase at the posterior. Then we consider another case where only the anterior third of the cornea is affected by cross-linking. So we have an increase of the stiffness only on the anterior third. And finally, we used another assumption where we we consider an increase of the stiffness in the anterior third, but in a way that evolves according to the time of the radiation. So the longer the time, the less deep is the penetration of the cross-link, and the higher is the increase of stiffness. So we used the second and the third model. The first one was not working to simulate the experiments. And actually, the second model, the one that accounts for an increment of the stiffness only in the anterior one-third of the cornea, worked pretty well for the cases with a low duration of the irradiation time, 5 and 10 minutes. Here you can see the preoperative behavior, then the application of the formula that we got through the model that we show, and these are the results for the postoperative behavior. For the 5 minutes, it's perfect. For the 10 minutes, we have some difference. For the higher times, 15 and 20 minutes, instead, the model that was really working is the post time dependent. You can see the green colors here that represent the 1/3 linear model, and the time-dependent model gets much closer to the experimental result. Actually, for 20 minutes, it seems to be the best approximation that we got. So using this model, we are able to predict the increment of the stiffness and so the result that you can have with cross-linking. Of course, this has been done with pig eyes. It's not something that is done on human eyes. Another observation that we get, it is a strict consequence of the fact that we are reinforcing, we are stiffening the anterior layer, is that the final distribution of the stresses changes very much. Here you see the normal stress in the nasal temporal section of the cornea. For the untreated case, the 10 minutes of irradiation and 20 minutes of irradiation. And you see that the level of the stresses grows very much because you have an increment of the stiffness in the anterior layers. Now let's switch to the simulation of transepithelial, LASIK, and SMILE surgery. As I said before, from the laser you can get all the information about all the layers that you can have in the cornea, but also the surgeons can implement inside the laser machine the surface of ablation for the transepithelial, and also the surface of cut or the surface of laser removal for the LASIK and the SMILE surgery. So since we have this geometry, we are able to model our corneas in order to simulate this kind of surgery. Of course, we had to do something because the LASIK and the SMILE surgery do not affect only the entire cornea, but only the central part. So we had to modify somehow our geometry in order to include a circumferential shape that represents the area of the action. Moreover, also in the thickness of the cornea, we have to insert to model directly the profile that is intended to be used for the LASIK, for the SMILE, and for the transepithelial surgery. So we have to modify our geometry to make them patient-specific, not only from the geometrical point of view, but also from the treatment point of view. And in the case of SMILE and LASIK, we had to introduce some numerical trick in order to describe the formation of the interface. Once you cut or you ablate with the laser the cup or the flap, well, you have an interface that you cannot eliminate anymore. It's well known that the lamellae, once they are cut, they cannot go back together. So we have a permanent interface within the cornea. So we had to model the interface, and with some experience inherited from cohesive elements, we were able to model the presence of a double surface that by doubling the nodes that belong to this interface, and keeping them connected until you are modeling the normal behavior of the cornea. After the surgery, instead the two nodes are considered as separated. They are not allowed to penetrate one within the other, so you have a kind of connector in the normal direction with respect to the stroma, but you have the possibility to slide along the tangential direction, because this is what the cup or the flap can do. So we did the model, and we had available the geometry of 40 cases, human, of course, with the perfect geometry for the preoperative cases. And then we model the transepithelial for all the 40 cases. And for the geometries that were apt to do that, we also model the SMILE and the LASIK cases. So you can observe that the average curves that reflects the IOP pressure versus the apex displacement in the preoperative case is characterized by a large dispersion. And also for the postoperative transepithelial, there is a large dispersion, but you can observe that the material is less stiff. In the smile in the LASIK, you observe that the behavior of the cornea after the surgery is more compliant, even more compliant. And again, you see in this plot the comparison between the average curves for the preoperative from the transepithelial, the smile, and the LASIK surgery. So smile and LASIK really weaken the behavior of the cornea, weaken very much the behavior of the cornea. I have to admit one thing, that in this case, we used for modeling the cornea, the human cornea, a uniform stiffness across the thickness, because the complication of the models were pretty difficult. So we didn't want to misunderstand something by inserting the fibers. So we did introduce the fibers, and we didn't account for the variation of the stiffness across the thickness. But the results are clear. In general, the most damaging procedure is the LASIK, followed by smile and followed by TE. And this can be also visualized in terms of displacements. These plots show the displacement in the direction of the axial optical axis. And in the preoperative, you have a distribution of the displacement that is pretty uniform. For the postoperative TE, the displacements are higher. But for the case of smile and LASIK, the displacements are not continuous. So we have a displacement jump across the interface. And here in this picture, you can visualize a little bit better the sliding of the cap and of the flap in the case of the LASIK, that show that the elements are not able to connect anymore in the same way that they were before the surgery. And the same thing, of course, happens also for the stress. If you want to compare the stress, you will get out with some similar results. So you will have a discontinuity of the stresses. These are the normal stresses in the nasal temporal direction. And you can observe that you have an increment of the stress in the posterior side of the cornea, so in the so-called residual stromal bed, in the case of smile and LASIK. And this hyper stress that is much higher may be responsible of the ectasia that may be observed in postoperative LASIK cases. Here, in a better detail, you can see how the cap or the flap for smile and LASIK, respectively, are less stressed, because since they are not connected anymore in a uniform way to the remaining part of the cornea, they do not contribute in a proper way to the stiffness, to the global stiffness of the system. Finally, I'm not sure about my time. Let me check. Okay, I think I will need 5 more minutes in order to conclude. The last thing I would like to show is related to the modeling of the keratoconus. So at the beginning, when I started in 2006 to study the cornea, I wanted to model keratoconus. And I started to use the continuum model. And I was thinking that in order to detect the shape of the conus, it was enough to reduce the material properties of the area of the cornea that is affected by the keratoconus. Unfortunately, this didn't work. Didn't work because the shape was not the classical shape of the keratoconus, and also the distribution of the stresses was not absolutely realistic. Something better happened a few years later, but actually the paper came out a year ago. And when we decided to use the patient-specific shape of keratoconic eyes. In that case, the results of the numerical simulation were pretty realistic. So the distribution of the stresses were understandable. And also we were able to reconstruct from the numerical point of view the refractive power maps for a keratoconic eye. Of course, we started from the correct geometry, and the refractive power maps are based on the geometry, so everything worked pretty well. But still, we were not satisfied because we were not able to model the evolution of the cones. And after a few years of attempts, we finally came out with some idea. Why we don't try to identify a micromechanical unit cell of the cornea that accounts for all the elements that are actually working in order to provide the stiffness of the cornea. So why don't we use the simplest model, a truss model, where we model as truss the collagen fibrils that form one lamella, the red cable here, or the blue fibrils that model the second lamella that runs orthogonal to the previous one. We model also the presence of some matrix, the embedding matrix, using these black struts that are in the radial direction. And finally, the cross links, so the bonds, the chemical bonds that keep together the lamellae, are modeled with other struts, with other trusses that are instead inserted diagonally. Okay, so this is the unit cell. If you modify this unit cell in order to get the shape of the cornea, you get a model like that. And of course, for numerical reason, the model has to be discretized in a very fine way. So what you see on the left is the model of the cornea, not the cornea, but the reinforcing structure of the cornea with a patient-specific shape. Now, if you apply the forces that are equivalent to intraocular pressure, in the case of materials that is healthy, so we assigned to all the struts the stiffness that was obtained from literature data concerning the collagen and the cross-links stiffness, you get exactly, if you put the healthy value, you get exactly the shape of the cornea under interocular pressure. But if you reduce the stiffness of the material gradually from the limbus to the center of the conus, you observe that applying the same intraocular pressure, you get the bulging. And the bulging finally has the shape of the conus. And this was very, very satisfying. Of course, the degradation of the tissue could not be a degradation uniform or concentrated. We have to have a law that describes the degradation. Actually, this is a parabolic distribution of the stiffness from the limbus to the center of the conus, and this provided this very nice form. The last thing that was missing is the thinning of the cornea. At the center of the cornea, we still have some thickness that was corresponding to the original one. So in order to resolve this problem, we thought that perhaps the cross links that are the bonds that keep the fibers together must be modeled with some more realistic chemical bond. So we made a resort to the potentials of Lennard-Jones type. And so the Lennard-Jones type potential are able to describe the weakening of the bond when the fibers separate. Moreover, we characterize this potential with a damage parameter. The damage parameter is a scalar parameter that we cannot identify right now how a chemical, a enzymatic, or other kind of damaging agent. But we assume that this variable that accounts for the damage diffuses in the cornea, starting from the center point of the conus, according to the results of a reaction diffusion equation. Okay, so we solved this problem first, and then the results in terms of distribution of these C variables were introduced in the discrete model of the keratoconus. And actually, we finally got something very interesting because we were able to capture the shape of the conus with also the thinning of the conus. So here I would like just to conclude with an animation that shows first the inflation with intraocular pressure, and then the time degeneration of the tissue with the deformation of the conus. And I repeat, with continuum model, this is almost impossible to me. I was not good enough to do something with the continuum models, but with this discrete model, we were able to do that. And here also, this animation shows the evolution of the axial stresses that you have inside every strata of the model. First, you see the reaction to the intraocular pressure, and then the effect of this damaging agent diffusion. So after having shown the results of our studies in therapeutic cross-linking in terms of experiments and numerical simulation, and modeling the results of transepithelial, LASIK SMILE surgery, and keratoconus degeneration, the research that we are trying to do right now is the modeling of the corneal lamellar transplant, the modeling of a therapeutic cross-link with the epithelium on, and we want to do experiments in collaboration with Cosimo Mazzotta on human corneas, and then modeling of retina degeneration. And I conclude with listing the names of the engineers and ophthalmologists that have been contributing to these studies. I'm going with alphabetical order. So Maurizio Angelillo, Alessandra Bonfanti, Federica Boschetti, Armand Cornaggia, Maria Laura De Bellis, Alessio Gizzi, Andrea Montanino, Eslini Annid, Paolo Sanchez, Irene Simonini, Marcello Vasta, Sanevo Verbeke, and the ophthalmologists Marco Ferrise, Cosimo Mazzotta, Giuseppe Perone, Tommaso Rossi, and all the members of the International Society for Eye Technology in Ophthalmology that has been supporting my work in this year. So I conclude, and I would like to thank again you for your attention. And if you have any question, please feel free to ask. Thank you.
Well, thanks, Anna, for a wonderful talk and some really beautiful results there. Really enjoyed hearing about all of that. So I haven't got any questions that come in from online yet, but if you would like to post them, then please do. Whilst I'm waiting, I've got a few of my own, so I'll just go through some of those until I see some from yourselves. So one question was, I may have missed this, in which case, sorry, but what was the technique that's used, the experimental technique that's used to measure the geometry of the cornea?
Well, it was an image segmentation. I don't remember. It was done with some commercial software. I think it's a JImage or something like that, but I don't remember, because this work was done by two master students, two girls, very nice, very good girls, because to take the images of 90 corneas is not an easy task. But they were taking the images at the undeformed configuration, so we didn't have the need to identify the unstressed configuration. And also they were taking the images at the final corresponding to intraocular pressure action. And that was a pretty big job, but it was done with some commercial software, and taking the images and getting the best fitting circumference that could get the anterior, the posterior shape, the thickness, the in plane measurements, and so on.
Okay, cool. And what's the physical device that's used to make the measurement? Some sort of tomography machine, I think you said.
No, it was a simple. It was a very high resolution camera.
Oh, okay.
Yeah, and so we had the laser that was measuring the apex movement, because this was the data that we were interested in order to calibrate everything. And the images were taken at every step of the pressure increase with a very high quality camera.
Oh, okay, cool.
Yeah, and we are recorded, of course. Everything was going in a list of in a sequence of images on the computer.
Okay. So another question, so some of the work you're doing here was with pig corneas. So I think, if I remember correctly, the size of a pig's eye is roughly the same as a human. I mean, how good a model for the human is a pig's cornea? What are the similarities or differences there?
Well, actually, let's say they are quite different. But among the animals, the pig cornea is the most similar to the humans. There are many, many differences. First of all, the pig cornea is oval, is not a circular, and they are elongated. So we had to modify our software in order to get the shape, not a circular shape. By the way, also the human cornea is not perfectly circular, but the elliptical shape is less evident. Moreover, the pigs they don't have fibrils that run according the distribution of the human corneas, because most of them they run circumferentially. So also in this case, as I told you in the simulation we did, we used a homogeneous material. We didn't implement the fibers because it was totally pointless to do something with the model of the humans when it was referring to pigs. So we use a homogeneous material, not an isotropic material, and which worked pretty well, probably. And let's say that the size is not too different from the human one. So they are a little bit larger, a little bit thicker. But if you take the mice, it's a total failure. Rabbit, total failure. They're completely different. Really, they are different materials. Also for cows, they are too big. They are completely, of course, they are very useful to understand the mechanism and so on, but you cannot use them to derive numbers that you can apply for the humans.
Sure. Okay, it's interesting. I'm surprised to hear they're so different. I might have just expected all corneas would be quite similar, but that's interesting to hear.
No. And also, if you think at the cats, they have this kind of pupil that are completely different from the human pupils. So every animal has a particular feature according to its survival needs.
Indeed. So I was going to ask, something I often wonder about with the treatments, these various laser treatments for the cornea. I think you also talked about a treatment where you treated with a chemical and then there was UV irradiation. Is there any is there any risk of damage to the rest of the eye or to the retina if you're using these sorts of treatments?
Okay, with the laser, of course, there is a big problem for the retina, but this new modern machine, they're able to control. They have some control system, so to avoid any kind of radiation on the retina. By the way, the laser is used also for many other surgeries inside the eye. For the removal of floating, for the fixing the retina when the retina detached. So you may need to use the laser to do other things. So the surgeon, the ability of the surgeon is really to use the laser in the proper way. For the UV, one of the reasons to develop faster protocols with higher power is really not to water too much the eye of the patient. At any rate, what is observed is that at the beginning of the cross-linking surgery, the UV radiation, they penetrate quite deep into the cornea, but then they don't go through. And the longer the radiation lasts, the shorter is the path of the UV radiation inside the cornea. So that's why we developed this model that was considering a reduction of the penetration of the UV radiation into the cornea as a function of the irradiation time. That seems to work. So let's say that in almost all the oxygen radicals that are form by the action, the collaborative action of riboflavin and UV radiation, they are consumed and they offer a kind of a block, a screen to avoid further penetration. But apparently, I know that the surgeons, they say, no, we don't have to bother the patient too much, but I never heard something like they are damaging the retina. So I wouldn't be, it's actually a pretty good procedure, a very, very good procedure.
Okay, cool. I wonder if I might ask a more technical question. So you're using some of the simulations. You're using a finite element method. I wasn't quite sure whether you're using quadrilateral or triangular meshes there, and if there was a reason behind your choice for this problem.
The cornea model has been developed by using quadrilateral blocks, bricks elements, let's say like that. And the reason was that we wanted to use the edges of the elements in order to define the orientation of the fibrils. If you look at the model, you have a transition. I can go back to the picture because perhaps it can be clearer. One of the slides here. Okay, this is okay. Let's you see that at the center, all the bricks are perpendicular one with respect to the other in the orthogonal direction. And then while you go to the boundary, they deviate from the orientation and they become radial and circumferential. Okay. Now in the mesh that you see here, we have some triangles. Of course, some of them may degenerate in 4 points. They degenerate in triangles, but it's okay. And for the SMILE and the LASIK surgery, we had to add some extra ring of elements because you cannot have, if you want to model exactly the circular shape, you need to have a circular surface. So we decided to keep the central part in order to have the orientation of the fibrils, and then to put some extra elements in order to be able to model the circular shape of the LASIK and the SMILE cut, incision, formation of the flap and on the cup.
Okay, that makes sense. Thank you. And I was wondering, is how computationally expensive are these simulations? So, for example, would it be feasible for clinicians to have some version of this code that they could run in a hospital, or is it too at the moment? Is it something that would have to be done on a larger machine?
No, I would say that since the model has some kind of microstructure inside, I'm speaking about the finite element model because there is this well-organized distribution. We observed that yes, of course, you have some mesh dependence, but the mesh dependence is not so strong. It's not that if you reduce to split by 2 the size of the elements, you get a much better result. The results are pretty. There is a very good convergence, let's say. So you don't need to have a very refined meshes. Something that sometimes I observe in alternative geometry of the eyes, they use a very, very fine meshes that probably are very expensive computationally. This mesh for simple cases, in the sense that when you're using a nuclear material, no fibers, it can take just 2 or 3 minutes to run. If you use the fibers, it may be 10 minutes because the model of the fibers with the dispersion, the variance, and so on is pretty complicated. But of course, most of the calculations we do are not parallelized because we run it on the fly, on the machine, and so on. But the code is all parallelized. So if you really want to do something expensive, you can do it in a parallel way. So you can have, we tested it up to 16 concurrent CPUs, but of course, it speeds it up, but it's not really necessary for most of the cases.
Sure. Okay, cool. Maybe I might ask one last question. So I think on slide 16, you talked about following treatment, you had an increase in the stiffness, but the dispersion of the stiffness decreased. I was wondering if you have any explanation for why the dispersion of the stiffness decreased.
Yeah, because you are reinforcing all the fibrils. Okay, let me go back because this was actually an observation that was done by my colleague Federica Boschetti, which is an experimentalist, and they say, look, I'm very happy to see this picture. Okay, you see when you apply some kind of reinforcement, the biological dispersions of the results reduces. So this is very interesting. The cross-linking really works. works not only in increasing the stiffness, but also making the outcoming material more similar. So you are kind of making uniform the material of the corneas that are treated with this kind of cross-linking.
Cool. Well, thanks ever so much, Anna, for a really wonderful talk, and thanks for taking the time to answer the questions there. We're approaching the end of our time now, so I'm just going to give a quick advert for the next month's talk. So our next seminar will be given by Tiernan Keenan on the topic of diverse applications of artificial intelligence and mathematical approaches in ophthalmology. And this will be broadcast live at 3 p.m. British Summer Time on Tuesday, the 6th of June. Further details of all future talks can be found on our worldwide page, the link to which can be found in the About section of this YouTube channel. And do also subscribe to this YouTube channel if you'd like to keep up to date on future talks. So it just remains for me to say thanks again to Anna, thank you to you for listening, and see you next time on mathematical and computational ophthalmology. Goodbye.
Thank you, Paul. Bye-bye.